Hello everyone. Hope all of you are safe and staying at home. Keep washing your hands. Okay. So in the last video we have seen about the paper pattern and topic wise weightage of science and technology part 1 2. Now let's begin with the actual syllabus. So here is the first lesson, heredity and evolution. So we know what is heredity. Last year we have studied about heredity and and variation. So heredity is nothing but the transformation of biological characters from one generation to the another generation with the help of genes, through genes. It is called as a heredity. So this is the we can see here that we look like our parents. So this is due to the heredity. And what is variation? So these are the dissimilarities which are found into the individuals. Okay, so we know that we look similar to our parents, but we do not look exactly similar to our parents. Okay, so the similarities which are transformed from the one generation to the another generation with the help of genes are that is the heredity, and dissimilarities which are found into the individual, okay, which are not transferred from the parents but they are got from the parents only, which are not present into the parents but they are found into the individuals. These are the variation, these are the differences, these are the dissimilarities. Okay, and the heredity and variation plays very important role in the evolution. Now what is evolution? Evolution is nothing but the gradual and the systematic changes which are taking place into the individual through the generation over a long period of time. So small changes which are taking place from one generation to the another generation, and into the certain different other changes are taking place from second generation to third generation, and this keeps on going gradually. Slowly changes are taking place, and at the end we can see very different organism or the different characters are seen into the certain generations, after certain generations. So this is the evolution. Okay, so before seeing all this, let's revise something that last year we have studied about the genes. So Gregor John Mendel, he has discovered the genes and characters which are transferred through genes. It was discovered by Gregor John Mendel that characters are transmitted from one generation to the another generation with the help of genes. So he is called as pioneer of modern genetics or father of modern genetics. So he has performed his experiment in 1886. Similarly, the research were taken place to study about the heredity in 1901. The reason behind the mutational changes or the sudden changes which are taking place into the individual, that is mutational theory of Hugo de Veris, which was reason behind the sudden changes were understood due to the mutational theory of Hugo de Veris. And meanwhile in 1902, Walter Sutton, here Walter and Sutton is given, but the correction which we have received from board, it shows that it is not Walter and Sutton but Walter Sutton. So Walter Sutton observed the paired chromosomes in the cells of grasshopper. So they observed the pair of chromosomes which are present in the grasshopper. Till that time it was not known that chromosomes are present and they are responsible for the transformation, they are responsible for the presence of genes, and they are responsible for the transformation of characters from one generation to the another generation. Also, so in 1944, the trio of scientists, that means the group of 3 scientists, Oswald Avery, then MacLean, McCarthy, and Colin MacLeod proved that except viruses, all living organisms has DNA as a genetic material. But later it was discovered viruses are also containing DNA. Now we are knowing the coronavirus, it also has specific coding of DNA and it keeps on changing. That means the changes in its DNA are taking place, so it is called as a mutation. Sudden changes which are taking place into the DNA is called as a mutation. In 1961, the French geneticists, that is Francois Jacob and Jacques Monod, they proposed a model of process of protein synthesis. So how the protein is synthesized into the cell, this model is proposed by these two scientists, French geneticists, that is Francois Jacob and Jacques Monod. And here this synthesis of protein into the DNA with the help of DNA and RNA, it gives a new field of genetics, genetic engineering and biotechnology. So last year also we have studied small part about the biotechnology, and in this year we will be studying some more part about the biotechnology. So the basic biotechnology includes the genetics. Okay. Now the very important process which are responsible for the protein synthesis is called as is nothing but the transcription and translation. So what is transcription, what is translation, what is translocation, that we'll be seeing. Before seeing that, let's recall about the DNA and RNA, because it is very important to know about the DNA and RNA before studying this transcription, translation and translocation. If you are not knowing about the DNA and RNA, then we cannot understand what is transcription, what is translation, what is translocation. So DNA, we know that Watson and Crick has given the model of DNA, which is double-stranded, helical and highly coiled structure. So on each strand of the DNA, the different nitrogenous bases are present. We know the full form of DNA, that is deoxyribonucleic acid. So the DNA molecule consists of deoxyribose sugar, phosphoric acid and nitrogenous base. So the strands are made up of deoxyribose sugar, and on that strand nitrogenous bases are present. So there are 4 types of nitrogenous bases which are present in DNA, that is adenine, thymine. There is a group. Okay, so adenine get attached with the thymine, and cytosine and guanine. Cytosine attached with the guanine. So on one strand, if there is a adenine, on the another strand there will be thymine. If on one strand there is a cytosine, on the another strand there will be guanine. Or if here it is guanine, on one strand it is guanine, on the another side it will be cytosine. Similarly, if on one strand there is a thymine, on the another strand there will be adenine. So it is a highly coiled double-stranded structure which contains the nitrogenous bases. Okay. Now RNA, and we know that also we know that DNA is present in the nucleus of the cell. Okay, into the nucleus of the cell DNA is present. Now this DNA forms mRNA. How it forms mRNA? Before seeing that, let's revise about the RNA. So there are 3 types of RNA, that is mRNA, tRNA and rRNA. So mRNA means messenger RNA, tRNA transfer RNA and rRNA ribosome RNA or ribosomal RNA. So tRNA and rRNA are present into the cytoplasm, that is outside the nucleus, and mRNA is produced into the nucleus on the DNA. After the production of mRNA, it comes out of the nucleus and it is also present into the cytoplasm. Okay. So in RNA we know that there are also nitrogenous bases. And which nitrogenous bases are there? Here adenine, guanine, cytosine and uracil. Here instead of thymine there is a uracil. In DNA, adenine and thymine are getting attached, they are getting combined, but in RNA adenine and uracil are getting attached or getting combined. So each of this nitrogenous base is called as a nucleotide. Separately, or if we will take it single nitrogenous base, if we will take, then each of the nitrogenous base is called as a nucleotide. So each of the nucleotide has its opposite or complementary nucleotide. Like we have seen now, in DNA, adenine combines with the thymine, guanine combines with the cytosine. Similarly, in RNA, adenine combines with uracil, guanine combines with cytosine. Okay, so now the production of mRNA, how the mRNA is produced into the DNA. The production of mRNA with the help of DNA into the nucleus is called as a transcription. Listen it again very carefully. The process of production of mRNA into DNA with the help of DNA. The production of mRNA with the help of DNA into the nucleus of the cell is called as a transcription.